I request all the participants kindly keep their videos off. Videos and microphones, please keep off.
Well, with permission of the organizers, I will start my lecture. And as you all know, that I'm going to speak on empirical data, its importance in research and analysis. So for this lecture, what I have done today is, compiled, the PowerPoint presentation with a view to, give you an insight of its application, because in one of the presentation, Dr. Kalam has very rightly said that in education, in our education system, what we do is give weapon to our students. I mean, it's like giving a weapon to students, but we don't teach them how that weapon is to be used. So now that on Google and on other platforms, a lot of information is available. So I have not gone into those details of giving you all that is available in public domain. What I have tried to do is from my own personal experience, from the research which I have done, I'm going to present you how that particular research project as an empirical research was conceived, and how I proceeded in conduct of that research, how the data was collected and how the data was analyzed. And also I'll also give you some example as to how this data can be analyzed. But since I have got huge data which I'm analyzing, but that will also become very complex. So what I will be doing today is give you few examples with very small data. But remember that a small data set can always be scaled up to say lakhs or million of data. And with computers available with us and good softwares available with us, it's very easy for us to analyze. But the issue will come when you have to do interpretation. So can I have next slide, please? You see, the first thing is we are using statistics in as a scientific method. And these scientific methods, start with framing question, as I have said earlier. This is something which I'll always insist when I was teaching students. I always used to tell them that there is no point only observing and recording your observation. And unfortunately, I find that most of the theses which are published, which I am able to see or which I had examined, were of observational type, where you do not start your research with asking a question. And when you ask a question, then you are very clear that you have understood the topic and you are exploring truth behind that question. And then after that you frame a hypothesis, and then you acquire the knowledge to prove or disprove that hypothesis. So that is how scientifically a research is done, and I will be giving you a few examples to explain how that is done. See, empirical means based on observation or experience. And here many times there are also problems, because people come up, because I do see in WhatsApp groups. There was one WhatsApp group where one gentleman posted that I have treated tympani and bloat by giving Dettol orally. And then when I questioned that, he said, no, this is my experience. So I'm not talking of that experience. Individual experience has no meaning. Your experience has to be in a very methodological, where when you put up an outcome, you will have to say that that particular outcome is not because of any chance. So statistics will always be talking about beyond chance. What is the probability that a particular outcome is a true outcome? So remember, so that is where in the outcome you'll have to come to. Then observation in empirical research, to collect empirical data is designed in a way to ensure accuracy, quality, and integrity of the data. And this is extremely important that when you look at any research paper, when you look at any thesis, you have to immediately understand whether the data that has been acquired is the correct data, and whether that data has been analyzed correctly. Because I remember there was one thesis which I examined, and in that thesis, they had done some work related with calf mortality. And in analysis, because I was the one who always used to even calculate. When a thesis comes to me, I used to calculate even all the calculation. I used to ask the guide and the student to provide me even raw data. And there I found that there was a problem that in calf mortality, instead of taking number of calves as the susceptible population, total number of animals were taken. So right kind of data is also important. Acquiring right kind of data is important, and its integrity is also important. Then another requirement of an empirical research is that any other person would also be able to generate similar empirical data. So your research has to be multiplied or must be replicated by others. Therefore, same observations or very similar observation you should be able to get at different situations and different locations. Scientific methods often involve lab experiment, and those are very easy because if you are doing lab experiment, much of the things are under your control, because you have everything which is calibrated, and there are only what desired variables which you keep. But once you go out, when you start observing nature's experiment, when you do collect data out from the population, there are so many things which are not within your control. So there are lots of variables which will come in. So collecting the right data and sorting out the variables which are not really required for your experiment becomes an important, and that also we would be talking in a short while. Then, for example, okay, how to identify empirical evidence. Now, because when you read a research paper, you have to really understand whether that is an empirical data based on which this research has been done and the interpretation has been made. And that holds good even for you, even for your students. Identifying empirical evidence sometimes be difficult, because what happens, the researchers, while publishing a paper, do not give details of the methodology. And there are a few points which may be critical, which are missed out. So those experiments then cannot be replicated. So a good research paper is one where all the experimental methodology that has been described is replicable. Some other person is able to repeat that experiment. And then it should have a detail of methodology. If you find that there are certain points on methodology which has been concealed and not described, then you can suspect that it is not an empirical research. And then definition of the group or phenomenon being studied. For example, if somebody is saying that I'm talking of retention of placenta, now in that case it should be defined that I'm talking of retention placenta where placenta has not been expelled even after 6 hours of calving, something like that. Then it becomes becomes very clear that the outcome is defined. Or you say ketosis where urine is positive for ketone bodies plus, and that the milk yield has gone down by 30%, 40%. So that definition of outcome is important to understand whether a particular research paper is empirical or not empirical. Then another thing you have to examine is whether that particular data is having any bias, and on that I'll be explaining to you in a short while. Scientists can be influenced by prior beliefs, and therefore, there are some misconceived ideas, especially this is very true with students, and therefore there is every likelihood that bias has crept in. So identifying, especially it is important for research guides who are participating today here. Identifying the bias which has been crept in is important, and that has to be eliminated before it is published. Can I have the next slide? I'll give you an example of, from my own research program, how a particular research is, developed, a research protocol is developed, how I ask a pertinent, empirical question. Now, this is the diagram which gives the flow as to how you start, how you start a research program. Now, the first thing is observation. You observe a particular phenomenon. For example, I give an example here of my own research program. I was getting lots of cases in buffaloes, and those were having coffee-colored urine. And this coffee-colored urine, we found out by doing diagnostic, serology, serum testing, was because of phosphorus deficiency. So this observation is, inspired me to look at, what is this problem, and then that is called as the induction period. In induction, you reason out that if it is coffee-colored urine and if it is hemoglobin in urine, then the RBCs must be, getting hemolyzed. And therefore, you, frame a question that, okay, how come phosphorus deficiency is leading to destruction in RBCs, so that hemoglobin is released and then hemoglobin is, excreted in urine, giving it coffee colored. And then you start, number one is, you deduce, from certain experiments. For example, you do for 20-25 cases and then say, okay, this observation that it is because of phosphorus deficiency is correct. And then, starts the testing phase. Now, in the testing phase, there are, I followed two methods, because the first thing was that, if I go to clinical cases, would have lots of variable, which I will not be able to control. So what I did, I formed an in vitro method, and in that in vitro method, I found out a technique by which I could only get the phosphorus out of the plasma, so that the plasma was absolutely phosphorus free. And then I added different levels of phosphorus, so that my first experiment was quite controlled. There were no variables. There were only one variable, that there were different varying concentration of phosphorus. And then I did a study to prove that, okay, what the hypothesis is correct, that phosphorus deficiency is leading to hemolysis, and the reason we found out. Then the second is evaluation. So in evaluation, we did a in vivo method out in the clinical field. Based on the in vitro research, we found out that how that phenomenon can be reversed. And with that formulation, we went out in the field, and then we tried that formulation and came back and said that, okay, this formulation works. So this is how the entire experiment was done. The positive point is, when you do this, not only you find out a solution to a problem, but you also find out if there is a solution which can be commercialized. And this particular formulation then was commercialized, by Intas, and this particular product is available in the market. So therefore, framing a good question, and then based on that question, having an experimental and clinical testing protocol is important to collect empirical data. And that empirical data then has to be analyzed properly to say that the results which you have got is true result, and it is not because of chance. Next slide, please. Another example I would like to give you, this is my PhD research program, wherein this I'm describing to tell you how a question is to be framed based on the available observation and available evidence in the literature. Now, I was working on trypanosoma, trypanosoma brucei. It's a blood parasite, which is there in animal as well as in human. In human, it causes sleeping sickness, and in case of animals, it causes SARA and several other diseases. Now, there was evidence available that this particular parasite is able to change its antigenic antigen, and that antigenic variation is because of what is called as a membrane surface glycoprotein. And an interesting evidence was observed by a USA group was that we know that the anchoring of many glycoprotein is because of amino acids. So instead of amino acid, here the anchoring was with dimeristic acid, to inositol. So dimeristate and inositol, dimeristate inositol. So here my question, which I framed for my PhD thesis, was as to how this dimeristate is assembled with the surface glycoprotein. I mean, what is the whole process of, number one, assembling of the meristate, and then transfer of this dimeristate, inositol dimeristate onto the surface glycoprotein. So with that question, I started working on it. And then another question which came into mind, because sometimes you also have what is called as a consequential hypothesis. That the first hypothesis is whether this dimeristate is inositol dimeristate is assembled and transferred, and if that is so, can I substitute the meristic acid with oxymeristic acid so that the whole structure is changed and this membrane glycoprotein is not able to enter. So I have an answer as a therapy. So that is how you have to frame question, frame hypothesis, and based on that hypothesis, then you can develop experiments. May I have next slide, please? Now, data quality, as I have said earlier, is important. If you want to have an empirical research, then you have to be very sure of data quality. Data quality depends on the right choice of variables and attributes. So if you are focusing on a particular outcome, then you will have to think of all the possibilities as to what kind of data I should collect, and that data should be directly associated with the outcome. So that will determine whether the data collected is correct or not. For example, I had said earlier that if you are collecting data of calf mortality, but you are including in the mortality rate, you are including the entire population, including adult, or if you're doing, say, for example, abortion rates, and in abortion rates, instead of including only breedable cows, if you're including all cows, then the data which you have collected is not correct. It's not the right kind of data which you have collected. If your focus is on early embryonal death, then you'll have to focus on only those cows which are in early pregnancy, and then from there you'll have to calculate what is the rate. So having right data and right variable is extremely important to determine the data quality. And an important question always keep in mind is, the more you have transcription of your data, there are more chances of errors. I have seen it myself that when you transfer data from paper to electronic, you have at least 10% errors creeping in. I'm talking of big data. I'm not talking of only data of 10 or 20. I'm talking of, say, lakhs of data from paper to electronic. So as far as possible, look at data if it is transcribed from the from paper to electronic. And if you are writing a research paper, and if you have transcribed data, then you please mention in your methodology that this data was transcribed from paper to electronic, and even from electronic to from one media to another media, that also need to be explained, because there are errors when you are doing this transfer from one format to another format. Now, data quality is a measure of condition of data based on factors such as accuracy. So the total quality of data will be determined by a number of factors. And you should keep in mind that data quality is important if you are going to take certain business decisions, because as I have said earlier, that I've got 1.2 million data, on animals data on my, and each animal is having at least 10 to 15 different types of data which is recorded. So you can imagine it's a huge data, and from this huge data, I'm also going to forecast certain things which are going to be used for business decisions. For example, I'm doing forecasting of milk being collected at milk collection centers. If that goes wrong, because of data quality, then the whole business decision is going to be wrong, and then they will run into crores of rupees of losses. So when it comes to big data, having data quality looked at is extremely important. Another example I can give is, you do progeny testing or genomic testing, and there, you have a reference frame which is, millions of, cows data. So there you have to be very careful that your data quality is good and it is not distorted. There is an example that, in IBM has calculated that annual cost of data quality issues in US amount to $3.1 trillion in 2016. And therefore, now we are getting into big data, and in big data, having quality. But fortunately now there are lots of softwares, that have come up, which can also look through your data and also point out if the data quality is good and acceptable or not acceptable. Next, please. Now, how to test data quality? So I'm giving you two examples over here, which will tell you whether the data quality is correct or not. And these are simple examples which I am giving, but these are true examples. You see, we do analysis of census, and whenever you have an official data before you, the first thing, because I wanted to publish a particular paper in a good international journal. So I knew the first question they will ask is, you are depending on official data, and what is the assurance that this data which has been collected, through official route is credible? So therefore, you have to also establish the credibility. So here I'm giving you an example of, how I have calculated for my article. I have calculated the credibility of the data. So what I did, I took data from 1997 to 2007 censuses, and then looked at number of percentage of animals in milk. Okay, because that data is collected in census. So you can see that's a fairly constant. So from 39 to 41%. So it's varying, very narrowly within 2%. So depending on trend, I would say that, because I know that farmers would always have in milk animals constant, because they don't want to have a large number of animals, extended population, because they have to feed them. So then I say, okay, now this is correct. My data appears to be correct trend wise. And then number two, I also looked at, what is the dry percentage, so that, breedable cow population, I could get that the range of, breedable population is varying narrowly between minus, 2% to plus 2%. Now, when I'm having, say, an X number of animals that are, in milk, they must have also given, birth to calves. And therefore, to prove that this number is correct, I looked at the number of calves, because in census, they also record the number of calves under 1 year of age. So probably, these are the number of calves which were born to these in milk animals at that point of time, with, plus minus, say, 2.5 to 5% difference here and there. So I looked at the number of female cows, because the male calves which are recorded is not under 1 year. They record, 0 to 3 years. So only I looked at the female calves, and then I proved that the data which I'm looking at, the census data, appears to be credible, because these are the quality testing which I have done. Another example which I'm giving you here is, for testing the quality of the data, you can also have some marker. And I introduce certain markers. When I tell students to go and collect certain samples, I also tell them to also collect the basic data, marker data of, that particular sample, so that then, you will be able to compare with that marker, whether that marker is fairly constant, and that marker has to be, something, which is present in all the samples which you are, sampling. So here I took two markers, in one of the studies, where it was an observational study, cross-sectional study, where sample data was collected. So I looked at age. I had three groups. So let's say in a control group, T1 group and T2 group, treatment 1 and treatment 2 group. So here I looked at the age. If I see that the age is fairly, the age average of the age, plus minus standard error is fairly constant, then I would say that the sample that is collected is representative. And you can see here that the T2 sample, the age is 5.2 plus minus 1.4. So that means the T2 sample that is collected is not really representing the control and T1, and therefore you have to go back and look at how the sampling for T2 was done. Similarly, another marker which you can look at, which I had looked at here, was average milk yield per day, because I was to study effect of a particular treatment on the milk production. So when I sampled 3 samples, then I should have all those 3 to start with, before I start my treatment. Their milk yield should be fairly within the range, plus minus within the range. So when you look at that will tell you whether the quality of your data, the quality of your number one animals you have selected is correct. And if the animal is correctly selected, naturally the data which will be coming to you is also going to be correct. Next, please. Now, sample size, when you are doing empirical research, this is also important. Many times I find that based on convenience we decide how much is going to be the sample size, or based on availability of funds to decide what is going to be the sample size, which is not correct. The sample size is always decided by the variation of that particular attribute in the population. If suppose you have got not variation is not very wide, it's just 2% or 3% variation, then in that case even if you go for a smaller sample size, it will be representative sample. But if variability in population is say plus minus 15% and 20%, then you need to have a larger sample size to be true to say that the sample which you have drawn is going to represent the total population. And a simple calculation which can be done is 4pq divided by L square. Now here P is probability, or you can say the probability. Here I am mentioning about probability of a disease. That will tell you, suppose you have a population where the disease is present only in 0.5% of the population, and if you want to find out that a particular population is having that particular disease or not, unless I look at a larger population, I won't be able to pick up a disease which is just 0.2 or 0.5%. But if I say that I have got a disease which is 25% in the population, then even if I take few samples, then I have a chance of having a positive case in my sample. So that is why the probability is important. Q is 1 minus P. So for example, here the 30% is the prevalence rate of say Brucella abortus detected by serology. So there is a area where Brucella abortus serologically was fairly common, 30% prevalence. And in that population, if I want to find out if there are cases of Brucella from serological basis, the calculation says that I'll have to collect 336 animals from the population to be sure that within 5% of the assurance that my error margin is not going to be more than 5%. I'll have to select 336 cases. So this is how you'll have to have a sample size. But this I have given you an example for a disease condition. There are also other examples which you can find out from the literature where straightforward formula is given, and then you have to fit in your variables in that, and you'll be able to find out how much is the sample size. So when you are doing research, please see that you stick to the sample size which is drawn empirically and is not based on whims or based on the convenience of availability of animals or availability of funds. Next, please. Metadata. This is something which is always talked about. When you are reading about big data, they also talk about metadata. Now, metadata is nothing but description of the data. Because when you are talking of big data, you also need to have a label of that data. That this is the data which has been generated during this period. This is data from cow. This is the data which has been captured during this particular period. Blah blah. All those details you have to save that so that after some time when you want to go back to that data, you know what exactly is the data and from where it has been drawn. So that is called as metadata. Next, please. And when you are dealing with big data, having metadata is important. Otherwise, you get lost, because after 6 months or 1 year, if you look at the data, it's very difficult to recollect from where the data has been collected. Now I come to the next point. What are the methods of collection of empirical data? Now with availability of lots of apps, computers, collection of data on paper is becoming redundant. And the simple reason is that if you are collecting data electronically, then it has got a lot of credibility. And number two, if data is collected and transmitted in real time, not written on a paper, but sometimes what happens, people write in pencil. Afterwards, they look at data and find out if some manipulation is to be done, then it is transcribed in ink, and that is maintained in the lab book, and from there it is transferred electronically. So there again there is a chance that somebody can play around. So if you do that, then it becomes difficult to establish that it's an empirical data which has been collected correctly. So this is one example I'm giving. This is from my own app, and this is the animal identification system which we have developed to increase the fidelity of the data. Now here there is a QR code, and anybody who is entering AI data, milk data, or any other data has to scan this and then enter the data and then transfer electronically to the server. Now this ensures that the person has gone to the animal, number one, because unless he is able to scan and his GPS is located in the server, that is verified, cross-verified, he is not able to enter the data. And number two, when he enters data from the animal side, there is no other transcription which is required. So that becomes a very credible data, empirical data which can be captured. Next, please. There are also now other IoT devices that are coming up, and the data which we are going to collect with IoT devices. Number one is real time, huge, and then it is collected dynamically. See, for example, now it is shown that when you take ECG of an animal or a lab animal, if there are people from pharmacy, they would be doing lots of studies in lab animals, and in lab animals they would be recording certain attributes after giving drugs. For example, temperature, for example, heart rate, for example, ECGs. Now generally what we do is we go to the animal once or twice a day, and at that point of time we collect this information. It is quite possible that at that point of time animal is not having fever, because the nature of the fever is diurnal. So therefore you need to record fever at a fairly constant rate throughout the day. Similarly, instead of recording ECG, when you are handling an animal, having an ECG recorded when animal is in its own natural environment is going to give you much more credible information than which you are going to collect. And there are also devices like you put a bolus in rumen. So instead of taking out a particular point of time rumen fluid and finding out its pH, you put a bolus with a pH sensor in rumen, and that will transmit data as per your system, that every 2 hours I need to have a pH. It will transmit data to your server through a transceiver. So now a lot of modernization is coming up in capturing of the data. And if you are using these electronic data which is properly calibrated, the credibility on the data quality is going to be very high, and therefore you would be able to classify such data which is coming from the animal side is going to be empirical and going to be useful to you. Similarly, there are devices like for clinical data. For example, you are interested in having heart rate, you're interested in having abnormality in heart sound. So there are large number of tele devices that are coming up, like tele ECG, tele stethoscope, and all these things are now available even for lab animals. So those friends who are from pharmacy colleges should know that there are now all these gadgets that are available. You have to just put a transceiver and you will be capturing data electronically. So you will have a lot of credibility on the data which will be collected. Next, please. this is another example where I'm participating in this particular research with Geo, Reliance Geo, where we are trying to develop a device, pedometer device, which suits to our animal's temperament, which suits to our ambient temperature and our management system, and also very cost effective. So again, here the entire animal health data, for example, what is the activity, how many times rumination, how many times it has eaten, how many times it was resting, how many times it was lying, all that health related data will be directly coming onto the server system, and server will recognize which is the normal data to be eliminated and which is the abnormal data which is to be kept and analyzed, and compare it with all other parameters, and then send to the clinician or to the experimenter if there is any abnormality, so that he can come and look at it. So there is a huge transformation of data collection that is taking place, and there the major emphasis is that data should be collected only once, and then it should be sent electronically to different stakeholders, and data should be collected from the point where you are doing an experiment, and it should come directly, and the point where you have collected data should be validated. For example, if you are collecting milk data, you have to be very sure that your person who is collecting milk data has really gone there, and he has not sat in the room and he has concocted the data. So lot of times these kinds of problems crop up. Next, please. This is just one more example I wanted to know. There is one company from Sweden which has recently developed what is called as electronic nose. And with this electronic nose, it can detect lot of volatile organic compounds which are related with metabolism. Okay, so you just put this gadget as an insert or on the body, and it will keep on detecting different kind of those organic compounds, and it will analyze. So it's like GLC. So miniaturized GLC, it will find out what is that compound, whether that compound is related to what metabolism, and then you will be able to have that data with you. So here transformation, as I have said, where the electronics is getting married to the medical science, and coming up with diverse devices. Next, please. This is the future of animal. You can replace mice, you can replace rat, rabbit, any animal over here. When you will be working, doing experiment, with these animals, they will be probably loaded with all kinds of electronics, and then the data which will be coming to you is going to be very credible, going to be in real time in a natural environment, and the data quality is going to be extremely good. So these are all devices which are available. So I think the future experimental animal which we'll be using will be fully an electronic animal loaded with all these electronic sensors. Next, please. Now, how to refine data? There are variables which are called independent variables and dependent variables. So you'll have to, before start of the experiment, when you will decide what kind of data you are going to collect, you will also have to assign what is going to be dependent variable and what is going to be independent variable. Now, independent variable, as the name indicates, it cannot be changed, such as age, sex, parity, etc. So it's not, you cannot manipulate it. So it becomes an independent variable. And there are dependent variable, whereas dependent variable where it is dependent. For example, if you vaccinate, then it becomes unvaccinated animals. If it is a milking animal, then machine milking animals, it becomes a dependent variable when you are using milking machine. If you don't use milking machine, it becomes a manual milking. So administration of drug or not administration of drug, again becomes a dependent variable. So you'll have to identify which are dependent variable, which are dependent variable. And then there is also another set of variable which is called confounding variable. Now these confounding variables could be both independent and dependent. It could also be affecting say your affecting your outcome. It could also affect other kinds of independent data. So identifying what are the confounding variables and adjusting for it is very easy. To give an example, suppose I'm working for mastitis and I have collected milk samples and found out that there are 3 different organisms which have been isolated. Now it is quite possible that 2 organisms may be confounding whether, but the effect is only for one. And therefore here you have to find out whether the other 2 are confounding variable or whether they are also responsible for the disease, and what is the effect if only one is there, what is the effect if there are 2, and what is the effect there are 3. So these are to be then adjusted using the mathematical model. I'm not going to take you into details of those mathematical models because there are a lot of videos also available. And if you go through those videos that are available, you'll be able to understand how the confounding is to be adjusted. But there is one simple method by which you can find out if there are confounding variables and what is the impact and effect of those confounding variables. It's called as adjustment by stratification. And I'll give you some examples of this kind of an adjustment where it could be direct adjustment or indirect adjustment, and this depends on the known standard proportionate rate. So I'll give you 2 examples to describe what exactly I mean by it. There is also another method which is called as a multi-factor adjustment model. It's called mental Hassel adjustment of data, where you suspect that there are more than 4 or 5 different kinds of confounding variables that must be confounding the true effect. For example, there are 5 different factors, and you feel that all those 5 different factors might be affecting the outcome, but out of which one is the true effect and the other 4 are just confounding effect. And you want to you want to adjust for confounding so that you will be able to find out what is the true effect of one particular variable. And here I can give an example of, say, you are doing for milk production, and you want to find out that if I give supplement A, it is going to have an effect on milk production. Now, milk production can be confounded by several factors. For example, parity. It could be the stage of lactation. It could be the feeding practices if it is not constant. If I'm going out and doing a cross-sectional study, so there are 4 or 5 different factors, confounding factors, which are going to have the effect of outcome. But I want to find out what is the true effect of my supplement. Then in that case, I'll have to adjust my data for all those 3 or 4 different confounding variables, and that can be done by what is called as mental Hassel adjustment. If you go to literature, you'll be able to get good examples of how to do both simple examples as well as large examples. In my software system, I have done all this internally so that the client is not bothered about all these things. All he has to do is just click, and after clicking with all the stratification adjustment of the data, he'll be able to get the true outcome, true impact of what he's looking at. Next, please. Now here I give you a simple example of empirical data. Now here the example I am giving is from Wayne Martin's book only. So somatic cell count. Now it was found out that the normal somatic cell count in milk has to be 200,000. So for my non-vet friends, I would just give an explanation that to find out whether there is a disease in udder in a milking animal, what we usually do is count somatic cells in milk. And if somatic cells count go up, then we say that there is a clinical or a subclinical problem with the udder. So here I'm giving an example where the elevated means more than 200,000 somatic cell count per ml of milk, and normal means less than 200,000 somatic cell count per. And then the data is divided for early, middle, and late lactation. And this is important to understand because based on this, I would be having a control strategy or the treatment strategy. So I'd like to find out how many are those cows which are in early lactation, middle lactation, and late lactation. So I do the stratification instead of taking the whole lot of observation. I do the stratification, and here I find out that in early lactation there were only 11% cases, which were having high somatic cell count, 35% in middle, and 79% animals that were in late lactation were having high somatic cell count. So the data very clearly says that the problem is beyond middle and beyond, and not much in the early lactation. And if that is so, then the literature says that if you have got high somatic cell count during mid lactation and late lactation, probably this is contagious milk. It is cow-to-cow transmission. And this cow-to-cow transmission then can be controlled by using teat dip. If it was early lactation, then probably it was due to, say, infection that has been contracted through dry period, and therefore you have a high lactation, high somatic cell count immediately after calving. Now, in that case, my strategy of control would be dry cow therapy. So this stratification also helps you in deciding on what is going to be your intervention, because remember, I'm talking of millions of animals, and therefore when I give this data and intervention strategy to any client, he is going to spend huge amount, and therefore that amount, then I will be able to also, after 6 months or 1 year, I'll also be able to find out what was the impact of this intervention. And therefore, for credibility, I have to be very sure that the intervention which I'm suggesting is based on the empirical diagnosis which I have made from the data. Now, the same data, when it is stratified for age, you can see that there are all three rate points. So which says that even in heifers, you have got 25% animals that are having elevated somatic cell count, which is not expected. It should be less than 5%. Whereas 3 to 5 years and 6 years up is 75%. So this is something which says that this is a chronic subclinical mastitis, and if the animals have been there in the farm, as the age is going up, the somatic cell count is also going up. Next, please. Another example which I can give is fertility management problem. So this is one problem where in dairy operation there were 1.5 lakh breeding cows, and this data I could pick up because the loss is going to be huge, because here the target was that calving to conception interval should be 90 days. I mean, that is what they decided, that we should have within 90 days, we should have 90% of the animals, they should be conceived. And what data suggested was that there was a gap of 31 days. So beyond, against the target of 90, it was 121. And look at the standard deviation, because generally we don't look at standard deviation or standard error, but that is extremely important. If the standard deviation should not be more than 20 to 25% of the mean. If it is more than that, it suggests that there is a lot of variability in the data, and therefore it suggests that it's a problem which is related with not all the animals but few animals. So it's quite possible that in that set of animals there are few which are normal, and there are few which are highly abnormal, distorting the whole mean, and that is why your standard deviation is way going variable. Now in that case, what next step I would do is pick up only those which are then abnormal. So leave out those which are normal. So with a mere click, I would say that okay, I would like to have those animals which are more than this standard deviation. Give me a list. So that list along with the farmer's name and contact number will come to me, and then I can analyze that data to find out what exactly is happening. Now in this case, different indices were calculated, and from the indices, we could pinpoint that calving to first breeding was the issue, and therefore heat detection was a problem. So if heat was detected, breeding will take place. So probably farmers were not detecting heat at the right time, and therefore here the decision is going to be first training the farmers or using some kind of gadget for heat detection, so that we are not dependent on the farmer. So stratification of data and looking at different indices, and also looking at standard deviation and standard error, will give you an idea whether it's a problem which is a common source. If it was a common source, I would think that it is because of environment, management, nutrition, water supply. So all those are common to all. And if it is not common to, then it is individual animal variation, and that individual animal variations data then can be stratified and then different variables then can be adjusted, and then I can pinpoint. Because if I have to pinpoint an intervention for say around 1 lakh animal, and if he's going to spend 50 rupees per animal for administering this intervention, then remember he's going to spend 50 lakhs rupees. And 50 lakhs rupees, if he's not able to get the outcome, then he is not going to be pleased with your system. So therefore, my data analysis has to be very, 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 very sound and on analytical grounds. And there, when I will communicate, I will also communicate that what is the chance that my calculations are correct, and what is the chance that I could have done some error also. So that also gives confidence to the stakeholder that I'm talking out on realistic term, that this is what is going to be the outcome. Next, please. So same data, let's say analyze to find out what exactly is the factor. And here we found out that the retention of placenta is the factor, because whenever retention of placenta was there, the mean was around 86. So very close to 100, very close to that 121 with a lot of standard deviation. You can see 48. So around 50% of the mean was the standard deviation, which was suggesting that within this population also, where there is retention of placenta, there are few animals which are having endometritis after retention of placenta, whereas few animals are not having endometritis, and therefore their fertility is not affected. Similarly, the data also shows that this problem is existing more in those animals which are high yielders, because in high yielders you can see 71 and 48, standard deviation is 48. So again, here there are few animals that are having this problem. So that is suggesting that there is a problem of peak yield management here. There is a problem related with energy. So we can then in depth look at those two factors and find out what exactly is the issue and do the correction. That is what is the strength of having data and correctly analyzing data, that with confidence we'll be able to tell your client what exactly is the problem, how much he has to spend, and what is going to be the impact and outcome in terms of monetary impact. Next, please. This is one very popular experiment that was done by Wayne Martin and Alan May. It's also called as Bruce County experiment. See, what used to happen, there was a lot of shipping fever problem, pneumonia problem due to Passerella hemolytica in calves that were coming to sales yard, because their system was that all those calves which are to be sold were brought to the sales yard, and from sales yard there will be auction, and then people will take, the other farmers will buy those calves for rearing. So in this experiment, what they did was they collected data from sales yard as well as from ranch on a number of animals that were positive for pneumonia and there were no pneumonia, and that was that is given here in the first table. So sales yard, 120 cases of pneumonia, 380 cases were not having pneumonia. So you can see incidence rate was 24% and 5%. So that tells us that okay, sales yard is the source of infection, that from there the disease is getting picked up, or that is the place where lots of those animals that are coming to sales yard is having this kind of pneumonia, either during the transportation or right from the farm. The second thing is they stratified it whether age has got any effect, and with that the data was it's called a stratification of data for calves and yearlings. So those were the calves which were less than 1 year old. Yearling means those were the animals that were more than 1 year old. And you can see here that the although the sales yard was the one which was sourced, basically the problem was with young calves. Young calves were the one which are having pneumonia. And I'm not giving you data here, but the third table, they showed that the problem was more in those in those areas in sales yard where yearlings and the calves were mixed together. I mean, they were kept together. So Martin found out a strategy of controlling pneumonia, wherein he said that okay, pneumonia can be controlled only by segregating young calves from yearlings. So don't house young calves in sales yard with the old cows, and therefore your problem of transmission of pneumonia to young calves will be so remember with this data, very simple intervention. I mean, no vaccination, no other administration of drugs, but a simple observational confirmation that if you have two ages of animals mixed together, the young ones having lower immunity is going to contract infection and suffer from the disease. So this is the strength of statistics, where very simple intervention can be found out if you have correct data and if you are able to analyze the data properly. Next, please. Then bias, how to control. This is something which is very important. There are a large number of biases which are described in literature. I'm not going to details of all those biases. I'll restrict to few biases which are important from the point of view of researchers. The observer's bias, or the research student's bias, or the researcher's bias is extremely important, because after all, the researcher is a human, and therefore we have certain misconceived or preconceived preferences. And this is very common with research students. I found that when we tell the student that okay, this is the drug and this drug is known to increase milk production, he has got a preconceived idea that when I use this in my experiment, it should come positive, because there is already a claim that it is increasing milk production, it is increasing fertility rate, whatever it may be. And with that preconceived, the bias comes in, and that bias then is reflected when the data is being collected. Because many students also think that having a positive result is important for my thesis to go through. You ask many students, they will always say that if I have a negative result, then probably the referee or even my guide is not going to be happy, because I'm reporting negative. But remember, even reporting negative result is also contributing in science, because when you report anything that is negative, the other researchers would know that work has been done on this particular issue and they have not found any effect. So from that point of view also, it's a good contribution. So therefore, research guide should also tell students that the integrity of collection data and doing experiment is necessary. And even if it is negative, he or she should not mind. It should be reported honestly. Now, how this can be controlled, how this bias can be controlled. So this bias can be controlled by blinding, what is called as blinding, so that the researcher doesn't know what is drug and what is placebo. And that is concealed in an envelope and only opened once the data analysis is done. Now, any experiment or any clinical trial which you read in the research paper, in the research papers or in the journals, look at whether there was blinding. If there is no blinding, then I straightway deduct 50% of credibility from that research paper, saying that there could be bias, and if that bias has not been identified and adjusted in the research papers, I would not have a lot of belief on that data which has been generated. So blinding is important, and in blinding, you should also have a control with placebo. So what I usually do in clinical trials, Santosh Shinde and all others from companies know that whenever I do any clinical trial of any product from the companies, I insist that it will be double blind and placebo controlled trial. And I don't have one placebo. I usually have 3 placebos, so that it is 4 different levels of treatment I'm giving. One is the true treatment with the true dose rate. Another one will be where bare minimum amount of drug. For example, if I'm using 10 milligram, I'll use 0.05 milligram as another placebo. And then I will have placebo, which is just inert, and pack it properly so that the researcher is not able to identify which is the drug and which is not the drug. And that increases the confidence of the confidence on your data. Then another thing to control is many times you also record subjective data, either yes or no, fed or not, given or not, behavior active, not active. That also gives you a lot of bias, because sometimes if he knows that I'm giving a drug, and this drug should lead to animal being more active. And if animal is not active while recording, because of that preconception, he'll record that it is moderately active, it is active. So there, so instead of having subjective data which is collected, give a measuring scale. You can have a scale that, okay, look for this, if it is this much score, look for this much of score, and then total score is given. So that also reduces a lot of individual variability. And that I had done with one experiment where we were doing California hemostasis test for somatic cell count, and I found that when we say +2, +3, +4, a lot of variation, individual to individual variation. So that has to be adjusted. Otherwise, if you are having 4 or 5 people working for your experiment, there will be a lot of variability, because of those things. Then selection bias is also important. Sometimes we tend to select, animals that are easy to handle. I go to a farm and they say, I need 12 animals for my experiment, and the people will bring those animals which are docile, because they're easy to handle. For them also it is easy to handle. So there becomes a selection bias. Now many times in research papers we do write animals were selected in a randomized way, but it's not really randomized. Randomized means you have a sample frame of all the animals, and all animals have equal chance of participation in the experiment. That is the definition of randomization. But how many of us do that? But while writing in the research paper, we very explicitly write the animals were selected randomly. Another thing is, at the time of describing the outcome, I'm taking animal in one village, and that too in one farm. But while interpreting the data, I would say that this particular disease in Maharashtra, this particular disease in India. You cannot extrapolate data drawn from one small sampling frame to an entire state or to an entire country. So these are some of the bias which have to be looked into. And now with big data, there are also data which has to be cleaned up. There are software which will clean up your data, and clean up means outliers will be eliminated on both the sides. This also sometimes introduces bias, because if you have got exceptional data which is actually giving you some very interesting observation, is being eliminated at the time of cleaning up. So these are some of the examples. So what I usually do is I insist on doing data as it is without cleanup, and also do with cleanup, and then compare whether I have done something wrong by eliminating or rejecting data which was otherwise useful. Next slide, please. I think I have 2-3 slides more, so kindly bear with me. This is one example I'm giving from my own research experience. That you need to give full freedom to the researcher which is participating, especially the students. And before they start the experiment, telling them the ethics of doing experiment, telling them having credible data is important, because you are contributing into science. And if you don't do that, there are lots of errors which creep in, and then those are reflected once the research paper is published. Remember, there are lots of research papers that are published are also withdrawn later on, because then there are very critically negative reviews on those research papers. I mean, labeling them as fudged data, and then you have to, and recently, if you have read in newspaper, where top institutes, were required to, withdraw the data, from internationally renowned journals. So telling your researcher that he is free and he should not think that he has to get a positive result. So I was doing it was a multi-institute project where we were developing nanoparticulate rifampicin, and that nanoparticulate rifampicin went through all kinds of in vitro. We had a cell culture model, then we had direct Brucella melitensis, Brucella abortus model. Everything was done, and we got very good results. Then we came to last stage, where it was to be tried in mice, infected mice. And then infected mice, I found out that even after 1 month, the research fellow was not coming back to me and giving me any input as to know what is going on. So I got suspicious. I called him, and then I told him, okay, show me the data, correct data. And I told him, okay, because we are developing a drug, you have to be very honest, you have to be with integrity. So very cautiously he told me, sir, there is something very weird that is happening. I said, there is what? He said that when I give our nanoparticulate rifampicin, for first 14 days, I don't see any brucella. But after 14 days, there is recrudescence, and in the treated animals, I am getting brucella. And he said, I don't know what is happening, but our all in vitro studies have shown that it is extremely powerful. It eliminates bacteria even inside the cell, in the macrophage cell line. So I told him, no, do not worry, repeat it 2-3 times. Every time, repeated same result. I went back to the literature and then found out that there is a crop of bacteria, a subpopulation of bacteria, which is not inside the cell, which is outside the cell. And what we were doing was with nanoparticulate rifampicin, we were delivering drug inside the macrophage, and there was very little drug outside. So what was happening? That small subpopulation which was outside the macrophage, we were not able to tackle. We were able to eliminate one which were intracellular, and therefore there was recrudescence. So then we came up with a hybrid, kind of formulation, wherein we said, okay, we'll have 3% free rifampicin and 70% rifampicin nanoparticulate. And taken together, when we administered this to mice, we got, total elimination. And even after 3 months, we were not able to see any recrudescence. So therefore, sometimes you get a negative result, but if it is properly and honestly, conveyed, then it adds into science. Otherwise, we would have never been able to develop this kind of concept, where you can have free as well as intact formulation for getting rid of infection, which could sometimes be outside the cell, and sometimes could be on, and number of times it is inside the cell. Next, please. Next. Yeah, this is another example. Where there is a reporting bias, there is a selection bias. That's doing nanoparticulate buparvocone study, and then the research fellow he presented the data, saying that when I use simple buparvocone, there is 35% recovery, 45% death, and 20% animals are not reachable. They are missing out from the population after study. Whereas he said that nanoparticulate buparvocone, 85% recovery, only 5% death, and 10% not reachable. Now remember, buparvocone is a drug which is already available in the market, and it's available, it's being used. So naturally, it must be effective. It must be effective, but it can't be 35%. So I got a suspicion. I said there is something wrong, here. So I told him, let's stratify your data to low severity, medium severity, and very severe. Because sometimes what happens when you're selecting animals, when you're allocating your animal to particular treatment, then you try to allocate subconsciously. I don't say it may be intentional. You try to allocate an experimental an animal which is under experiment to a group which is preferred by you. So less severe, will go to my preferred treatment, then the second one, and then the third one. And this was not a blind trial, so because I cannot do blind for a clinical case with owner. So I cannot tell him I'm going to give a placebo. That is unethical. So this was not blind. So the researcher knew which was the drug and which was not the drug, because he had also to do mixing at the field. So he had all the chance to know which is the drug, which is the nanoparticulate drug, and which is the normal drug available in the market. So I told him to stratify, and then you can see from stratification, you can see here the problem was that more number of cases in case of BPQ were allocated to more severe cases. Out of 100, 33 cases were allocated to severe animals, and therefore the mortality rate was very high. And then I told him, okay, no, you go back and redo the experiment, redo the clinical trials, and there you do what is called as matching. Now, matching is also a very strong, way to eliminate bias, selection bias. Where I gave him the range. For example, it's hemolytic anemia, and therefore I told him, okay, we score the animal for different severities, low, medium. If you get one case which is low, then one low case goes to buparvacone, another low case goes to nanoparticulate bone. You get another medium case, it goes to buparvacone, the next will go to, so like that, what is called as matching. So here you both the groups are matched, and therefore they are in the equal standing, and the data which you'll get from matching is going to be more powerful to be convinced that your hypothesis and your formulation is really doing what you are claiming. Next, please. Thank you.
Thank you very much, sir. From participants, if having any query, please write down in chat box.
Now, people are asking for sharing of the PPT. So, Dr. Sajid.
Yes, sir.
You can share my PPT. There's no issue.
Okay. Yes, sir. Those who are asking will share at the end. Yes, sir.
And also show, they are asking.
Even we have a recording, sir, for this lecture. So if possible, we'll share the link for that lecture also after uploading on the YouTube.
There is one question somebody has asked: field ways to collect good data, as you earlier said that we don't. Can you read, Sajid? That in the chat there is one big comment somebody has.
Wait a minute, sir. Wait, sir.
Now there is one question where you said that 4 PQ upon L square. Please, he said, you have to, for elaboration. So here, 4 is, approximation of Z value. Okay, so 1.84. So it's a square approximation taken as 4. P, as I've said, is probability of, having that particular, outcome in the population. For example, here, I have said, 30%, there is 30% prevalence of, this particular disease in the population. So here P will be taken as 0.3, and Q is 1 minus P. So that will be 0.7. Now L here is what is the error margin, you would like to be. So this error margin could be, say 5%, 10%, 20%, depending upon how critical, is your experiment, for policy making. So generally, we do such kind of studies with 5% to 6% error. So L will be then 0.05 or 0.06 square. And if you do that calculation, you can find out what is the number of the sample size number. And so, for example, here I had given you that 336 animals are to be selected, as sample size. Suppose it's a farm, and that farm is having now only 200 animals. Then how can you select 336 animals from that farm? So in that case, there is another formula which is called 1 upon n plus 1 upon capital N. So 1 upon capital N is the number which I have drawn, like 336, and 1 upon n is 1 upon 200. That is the actual population of the farm. And if you do that, you'll find out how much from 200 it needs to be selected. So these are I've just given you an example. You can go back to Google. You will get all those details, but only to understand how it is to be applied. So whenever you are selecting a sample, that sample should not be based on convenience. It should be based on some scientific methodology, and that methodology, when you are publishing the paper, should be described.
Sir, one question is related to the policy actually.
Yeah.
He is asking that this is the age of big data in veterinary science. We are generating huge data daily in field, in lab, in pharma. But it's heartening to know that what he's saying, MAPSU Nagpur doesn't have separate department for livestock economics, statistics, and IT.
Okay.
So
Actually, friend, it's a good question, he has asked, because this will have, lot of implications. When we look at, I mean, and I don't want, I mean, it's a criticism, of course, and being an academician, I have to be very critical. This is something which I would like to, mention for all my friends, who are in faculty. You see, when you are in academics, you should have lateral thinking. I mean, you should look at anything, everything that is presented to you from different angle. And that is what, that is what I have been doing. If somebody says, this is the common, popular, policy strategy, I look at this, very critically to find out whether it's a populism or it's a market strategy, where there is a marketing company which has come up with a scam, and therefore this is being popularized. But the problem is generally the academicians go by what is the general flow. I have given an example of that sex semen, and nobody has done any study. Nobody is coming out and criticizing that policy, that it's going to play havoc. In the beginning, when crossbreeding program was implemented, nobody critically evaluated how it is going to impact our own indigenous breed. Whereas we went to Canada and we went to other countries, and breeders of those companies, because I happened to visit, Holston Canada office, interacted with very senior, one fellow was 85 years old. And he told me that, in the meeting where Manibhai Desai participated in, that, in the meeting where this breeding program was being finalized, the breeders there was insisting that you have got a good quality cows, and you should not go for crossbreeding. You should go for pure breeding. Try to upgrade the genetics of your own animal, but you wanted to have a shortcut, and therefore we went to it. And now you have all kinds of criticism after 30 years or 40 years. So same thing is going to happen with semen technology. Our problem is somehow I find that we don't like having data, and that is because data gives accountability and transparency, and therefore we would like to have a smoke screen and tell people that everything is fine. I can tell you that there is a large resistance in collecting an analysis of data. No, the NDDB has started a program. Remember, what kind of problems you are facing. Now, I don't think there is. There must be one or two colleges who are even maintaining their own farm record. I have collected data under RTA from some state governments about their farms, and 60 to 70% of the farms are communicating that they don't milk cows. So leave aside, giving you record of milk. So they don't know whether there is genetic improvement, but the entire farm is getting sponsorship for genetic improvement of this breed and genetic improvement of that breed. But how much genetic improvement you have been able to do after 50 years? Have you done any study? They say, no, we have not done any study, because there is no data. So you can't do any study. So this is a very big problem where everybody has to think. Creating a department is fine, but unless that department is going to have access to large data, and that large data will be accumulated only when everybody participates, every single animal is identified. We know how mockery, we play with the policies. There was a policy that animal identification will be done and the data will be collected. But ultimately what happened? The livestock development departments purchased huge numbers of ear tags. I don't want to give you data of individual state, because I have access to all that data under RTI. Huge numbers, and very few animals were actually tagged. And these tags were purchased because it's very easy to purchase tag. Just place an order, tender, place an order. So we need to really sit up and have scientific temperament. That's what in my inaugural speech I had mentioned, that will come only with that temperament, where your thinking will be rational and you will rely on data.
So most most of the participants, they are asking about the outliers. One question is outliers in case of small animals, as different parameters like weight, fasting, glucose differ very rapidly.
Okay. I mean, that is what then, you'll have to adjust with your, I mean, if you are having correct randomization or selection of data, then you won't face any problem even with small animal data, because what outlier is, if you see that, standard curve, that bell-shaped curve, the peak, if you draw onto the x-axis, that is the mean. And then from mean on both the sides, you have one standard deviation, two standard deviation, on your left side of the mean, on your right side of the mean. Now, the outliers will be that data which is lying either beyond 3 standard deviation or 2 standard deviation on both the sides. So that decision rests with you, that, which are the outliers, you would like to eliminate. And if you are eliminating outliers in your experimental protocol, you must mention that the data was refined and the outliers were rejected. And as I have said many times, the outliers also give you very interesting insight and sometimes very novel insight. So therefore, I do both the calculations where I do not eliminate outliers, consider them, and then also eliminate them, and then find out whether I'm doing right. So that is how it should be done. So outlier means, compared to mean. So if you are having a sample population where there are young, medium, as well as adult, your mean is going to be different. So what here you are trying to do is that you should have around 62 to 63% of your population which is selected around the mean, one standard deviation, and then around 85-86%, 2 standard deviation. That's how, the distribution of the bell shape takes place. So you don't want to, go to outliers, because they will severely distort, and therefore your statistical significance will go down. And that usually happens when you go out in field and have observational study and collect data, based on some physical information. There, you have, this problem. But in small experiments where, you're doing lab experiment study, this is not a problem, because here you are dealing with a very fairly controlled study where all other variables are controlled except the variable which is of interest to you.
Okay, thank you, sir. Let me inform the participant that we are having some pure statisticians, and most of their questions related to the standard deviation, standard error, and outliers will be covered by them. So I extend my sincere thanks to the our first speaker, Dr. Abdul Samad sir. He has given his valuable time to us. And the next speaker, Dr. Santosh Gite, is ready. So, sir, thank you very much, sir.
Thank you. Thank you. Good luck to all of you.
Thank you, sir.
Good luck to all the researchers. I hope